Hello, everyone, and thank you for joining me today. My name is Madeline Moffett, and I am a volunteer undergraduate research assistant here at the University of Iowa in the EDAL-ABLE laboratory. And today I will be discussing an ongoing project in our lab entitled Investigating the Potential Compensatory Relationship between OPA-1 and estrogen in platelet mitochondrial complexes. And before I get started, I would like to just take a moment to thank those individuals who made today's presentation possible, starting with my fellow undergraduate research assistant, Arnav Umesh, Dr. Eric Weatherford for providing the RNA sequencing data, my lab mentor, Dr. Rhonda Souvenir, who has been an incredible resource and has a tremendous impact on my career and also personal life. And of course, Dr. Edale Abel himself for providing me the space to continue to explore my love of science. And to get us started today, I would like to introduce Optic Atrophy Protein 1, or also known as OPA-1, which is an intermitochondrial membrane protein that is responsible for mediating mitochondrial fusion, as seen here in the diagram on the left. In addition, Optic Atrophy Protein 1 helps maintain cristae and mitochondrial morphology and oxidative phosphorylation, and does have implications in apoptosis. And what was found in previous studies was that an upregulation of OPA-1 was associated with increased risk of cardiovascular disease and thrombosis. So what we did in our lab was we performed platelet-specific deletion of OPA-1 in a mouse model. And what we discovered was a sexual dimorphism in terms of thrombosis. And the results from that study are summarized here in the table on your left. Starting with our males who were devoid of OPA-1, we actually saw compromised cristae morphology in addition to increased platelet activation and shorter time to form an arterial clot. Whereas in our female mice that did not have OPA-1, we didn't see any significant changes in mitochondrial morphology. Again, no phenotype change in platelet activation. However, we did see increased time to form an arterial thrombus. That prompted us to perform adaptive platelet transfer, in which we took male platelets and put them into a female host, and female platelets and put them into a male host. And what we discovered that these male platelets and female hosts mimicked the female phenotype. However, the female platelets in a male host did not show any significant changes in arterial thrombosis time or phenotype. This prompted us to start thinking that estrogen played a pivotal role in thrombosis in our females. So we then went ahead and performed gonadectomies to look at this further. So we removed the testes from our male mice and we removed the ovaries from our female mice. And what we found in terms of mitochondrial morphology was gonadectomized males did not show any significant changes in mitochondrial morphology. However, we saw compromised cristae morphology in our gonadectomized females. Again, we looked at arterial thrombosis, and in our gonadectomized males, we saw increased time to form a blood clot. However, we saw no significant change in phenotype in terms of arterial thrombosis in our female mice. So to look at the molecular changes going on in our female mice a little bit more closely, we looked at RNA sequencing data, which can be summarized in the figure on the right here. I would like to draw your attention specifically to the oxidative phosphorylation pathways, where we can see that we found upregulation in our knockout sham females. So our females that did not have OPA-1 but did have their ovaries and therefore had estrogen. In addition, we found upregulation in this pathway in our wild type OVX females. So our females that did not have OPA-1 but that had OPA-1 but did not have their ovaries and therefore no estrogen. Looking at these oxidative phosphorylation pathways a little bit closer, we did find upregulation of 26 nuclear encoded mitochondrial transcripts in our female knockout shams, and that can be found here in this middle panel. And in addition, we did see upregulation of these nuclear encoded transcripts in our wild type gonadectomized females, not to the same extent as our knockout sham females, but upregulation nonetheless. We then became interested in looking specifically at mitochondrial complex expression. So we performed blue native page, which can be seen here in the figure on the right. And what we found was a visible upregulation in our OVX control and our OVX knockout females specifically in super complex S1, which is the combination of complexes 1 and 4. In addition, we saw visible upregulation in our OVX females in complexes 1, as you can see here, and also in complex 5, as seen here and here in this panel as well. And in future research, we would like to expand our value of N and to add more samples into our experiments. In addition, we would like to next look at mitochondrial complex activity as well. But at this point, we do believe that in combination, the previous research that's been done and the research that we have completed, we do believe that there is strong evidence to support a potential compensatory interplay between OPA-1 and estrogen in terms of thrombosis in our female mice. And with that, I will go ahead and finish up my presentation. Thank you very much for joining me today.